Hello, dear students. So now today we would be discussing the remaining topics of the chapter, that is microorganisms, friend and foes, class 8 science. Okay. So and in the previous video we have discussed about the diseases in human beings, the different types of deficiency diseases we have discussed there. So now still you can see here animals are also susceptible to the diseases. If you could see the diseases which are found there in animals, like the disease that is most common in animals, that is the anthrax. This is the most common disease which is found in animals and is caused by, this is the disease which is caused by bacteria. Okay, this it's a bacterial disease. Okay, at the same time one more disease there is common in animals. There at that time there you can see, this is the foot and mouth disease. Okay, so one more disease, that is foot and mouth disease. This is also very common in animals. So this foot and mouth disease, and this is caused by virus, so it's a viral disease. Okay, so these are the two most common diseases which are found in animals. Okay, at the same time the plants are also susceptible to many sort of diseases. So if you talk about the disease in plants, so here I'm writing it, diseases in plants. If you can see the diseases that are in plants, that is, the first one is the rust of wheat. It is most common disease. It is caused by the, basically, the fungus, caused by fungus. Okay, you can see some fungus on wheats there, so it is also known as rust of wheat. Okay, so second one, you have the citrus canker. This is the second one, that is the citrus canker. So that is the most common disease which is caused by the bacteria, and this disease you can see mostly in the citrus fruits. Okay, uh, that is caused by the bacteria, and this is the disease which is commonly found in the citrus fruits, like lemon, like oranges. So these are the fruits which are susceptible to this one disease, that is citrus canker. Then one more disease is there, that is commonly found in bhindi, also known as the okra, and that is the yellow vein mosaic. Okay, that is yellow vein mosaic. So yellow vein mosaic of bhindi, also called okra. So this is the disease which is found in bhindi or okra, and caused by virus. So it's a viral disease. Okay, so these are the certain diseases in animals and the plants. Okay, so to protect the plants from these diseases, what do? We use the pesticides, we use the chemicals to protect the plants from these kind of diseases. Okay, now if you have written this, let's move ahead and let's discuss some other important topics of this chapter. The first one is the food poisoning. Okay, you might be knowing about the food poisoning food food poisoning. Okay, so what is this food poisoning? This is due to the consumption of the food that is spoiled by the microorganisms or the microbes. Okay, suppose you have a raw mango here, just you keep it out for certain days and then you are consuming it, so it leads to food poisoning. Why? Because there might be the attack of bacteria in that one fruit. Okay, so if you are consuming a spoiled food that has been spoiled by the microorganisms, so it leads to the food poisoning. So it is due to the consumption of the food that is spoiled by the bacteria. It is due to the consumption. I think it is not visible here. Let me write it downside. It is due to the consumption of spoiled food by microbes. Okay, so the food that has been spoiled by the microbes, if you are consuming that food, it would lead to food poisoning. Are you getting this? Yeah. So this is the food poisoning. Now, to preserve the food, okay. Suppose you are going to preserve the grain that you have stored. Okay. Suppose you want to preserve some food like meat, fish, etc. These things you want to preserve. Suppose you want to preserve the pickle. Okay. Suppose you want to preserve the jam, squashes, etc. So these things are to be preserved. Okay. So to protect the attack, to protect the food material from the attack of these microorganisms, which causes the food poisoning, so there is the need of the food preservation. Okay. So now the very important term, that is the food preservation. So what is food preservation? See, there are many methods to preserve the food. Okay. To preserve the food from the attack of the bacteria, or to preserve the food to inhibit the growth of bacteria in the food. So there are some methods to for that. So the first one is that is the chemical method. Chemical method. So certain chemicals are used, which are edible chemicals. Those chemicals which are authorized by the FSSAI organization are the edible chemicals. So these edible chemicals are used to protect the food. Okay. So certain chemicals are there, like you have sodium benzoate, like you have sodium metabisulfite. So these are some chemical preservatives. So these are used to preserve the jam, like these are used to preserve the squashes. Okay. So here you can write preservatives like sodium benzoate, we are using it. And at the same time you have sodium metabisulfite, also sodium metabisulfite. So these are some chemicals on, and these are the edible chemicals. If these chemicals are reaching in your body, so no harm is there. Okay. Because there are edible chemicals, these are authorized by the FSSAI organization. So these chemicals are used as preservatives. Yeah. Preservatives like these and this are used in case of, are used to preserve, like we are using it to preserve jams, we are using it to preserve the squashes. So these things are used to preserve these jams and the squashes. Okay. So this is the chemical method. Now some home method also we are using, like we are using the salt, we are using the sugar. Why do we use this? Okay. So salt is most commonly, it is used at our homes. It is used to preserve the meat, it is used to preserve the fish, it is used to preserve the amla, the raw mango. So these things are to be preserved by the use of the salt, because salt inhibit the growth of bacteria in the food items. Okay. So salt is used. Second one is salt is also used to preserve like meat, to preserve, we are using, with the help of this, we are using it to preserve the meat, we are using it to preserve like fish. Okay. Okay. Like we are using it to preserve amla, etc. So these things are used, so this salt is used to preserve these things. Okay. So this is the second one. Now one more is there, that is the sugar is also used as the preservative. Okay. These are the chemical preservative. Salt is also a preservative. Now one more preservative that we are using at home, that is the sugar. Okay. The sugar is what, basically the sugar reduces the moisture in the food, and due to this it inhibit the growth of the bacteria. Okay. So we are using the sugar for that also. So sugar, it uses, so sugar is used to preserve, like we are using it to preserve jams, we are using it to preserve squash. So these things are, so here sugar, we are using sugar to preserve these things. Okay. So why do we use sugar? Because sugar just it reduces the moisture which is there in jam and squashes. So it reduces the moisture, and due to this it inhibit the growth of the bacteria. Okay. One more thing we are using at home, that is the oil and vinegar. Oil and vinegar. So oil and vinegars are also used as the preservative, because there are some bacteria which cannot grow in the environment which is provided by oil and vinegar. So the environment which is provided by, we are using this oil, these things are using, we are using it in pickle also. So they are providing such an environment which inhibit the growth of the bacteria. Okay. So bacterial cannot bacteria, some bacteria are there which cannot live in the environment which is provided by oil and what can I say, this vinegar. Okay. So bacteria cannot live in the environment provided by oil and vinegar. Okay. So here oil and vinegar, these things are also used as preservative. One more is there, that is this hot and cold treatment. Next is hot and cold treatment, because basically if you heat something, if you boil something, it kills the bacteria, and if you cool down something, it again there are some bacteria which cannot thrive such a lower temperature. Okay. So boiling kills many microorganisms, and the lowering of temperature inhibit the growth of microorganisms, because if the temperature is very low, so the bacteria cannot thrive in such an environment. Okay. So boiling kills the, here I'm writing, if it is visible to you, yes, boiling kills the bacteria, and lower temperature inhibit the growth of microbes. So there are some microbes which are just which needs a proper temperature for their growth. Okay. But if you lower down the temperature, so the bacteria cannot thrive in such an environment. Okay. So this is how hot and cold treatment is also useful, and it is also used as a technique of for preservation. Okay. Now one most very important technique, that would be the sixth one now, and it is the pasteurization. Okay. So if you have written this, let me erase this. So this would be the sixth one now. Sixth technique, that is the pasteurization. It is especially in the case of milk. Pasteurization. Okay. Especially it is done in case of milk we are using. It is done in case of milk, especially in case of milk we are doing. What we are doing here, we are here the rapid heating and the rapid cooling, these both takes place simultaneously. Okay. Rapid heating and rapid cooling, this process takes place here. So this leads to the pasteurization of the milk. Okay. Here rapid heating, and here I'm writing, here rapid heating and rapid cooling leads to pasteurization of milk. Okay. Rapid heating and rapid cooling. So at first there will be the rapid cooling. Okay. At first there will be here lower temperature. So at first there will be the rapid cooling, that will be lower temperature, and it would be around you can say 60 to 62.5 degrees Celsius for 30 minutes. So for 30 minutes, we just lower the temperature, and around the temperature would be around 62.5 degrees Celsius, and it is for 30 minutes. So for 30 minutes, we lower the temperature. Then suddenly we increase the temperature for a short duration, that is around 15 seconds. Okay, so this leads to pasteurization then. Okay, and then higher temperature, and it is around 72 degrees Celsius, and it is for 15 seconds. So you can see higher temperature is for short duration and lower temperature is for long duration. So this will lead to the pasteurization of milk. Okay, here for long duration, you just keep something in the lower temperature, then suddenly you raise the temperature up to 72 degrees Celsius, and it is for short duration, that is 15 degrees Celsius, and then it would lead to the pasteurization. So this is especially we do in the case of the milk. Okay, one more technique is there, that is you just storage and packing. This is also a technique for preservation, storage and packing. So this is also done. Suppose if we keep something in the airtight packet, so this is how also we can preserve something, and we can preserve it from the bacterial attack. Okay, you can write here. It can be done in airtight packet. Airtight packets. You might have seen something you are getting from the store like Kurkure, etc. These are kept in the airtight packets. Okay, why do we keep them in the airtight packet? Because to preserve it, and to preserve it from the microbial attacks. Okay, so that is why they are kept in the airtight packets. So these are 7 techniques for the preservation of food. Okay, then the last topic which we have to study here, and that is the nitrogen fixation, that we have talked already in this chapter, that is the crop production and management. There we have already talked about it, that is the nitrogen fixation. So this can be done by a bacteria, that is the rhizobium bacteria, which is found in the root nodules of leguminous plants, helps in the fixation of atmospheric nitrogen. How it is being done? Now see, suppose in atmosphere, if you see, there is 75 to 78% around, there is nitrogen in our atmosphere. Okay, this nitrogen is necessary for the growth of the plants, but the plants which are here, these plants cannot take this nitrogen from the atmosphere. Rather, this nitrogen needs to be converted into the nitrogenous compounds, like it needs to be converted into nitrite, it needs to be converted into nitrates. Okay, so this nitrogen needs to be converted into nitrite and nitrate. Okay, so how it is being converted? So this is called the fixation of nitrogen. Okay, so nitrogen is being converted to nitrite and nitrate. So this is called the fixation, and this fixation is done by a bacteria known as the rhizobium bacteria, that is found in the root nodules of the leguminous plant. In these root nodules, these rhizobium bacteria is found here. This rhizobium bacteria helps in the fixation of this atmospheric nitrogen, and it converts this atmospheric nitrogen into nitrite and nitrate, and then it dissolves into the soil, and later it reaches into the plants. Okay, so this is the function of the rhizobium bacteria, and this is how the nitrogen fixation takes place. Okay, this we have already talked. Okay, this is how now we are going to conclude this chapter. Would be discussing in the next lecture the next chapter of science, that would be the third chapter. Okay, till then keep stay tuned and keep watching. Thank you, have a good day.